Good day, everyone, and Assalamualaikum. First of all, I would like to introduce myself. I'm Dr. Khairul Fairuz Azman from the Department of Physiology, School of Medical Sciences, University of Science, Malaysia. Right, so in this video, we are going to talk about renal physiology, specifically the structure of the nephron, its blood supply, and one of the most important functions of the kidney, which is glomerular filtration. So by the end of this video, you should understand the structure of the nephron, renal blood supply, or how blood flows through the kidney. And you should also know how glomerular filtration occurs in the kidney, as well as glomerular filtration rate, or GFR. So this is the gross anatomy of the kidney. If we look at it from the outside and then cut it open, we can divide it into three main regions: the cortex, medulla, and the renal pelvis. So the cortex is the outer regions. It appears lighter in color. So this is where glomeruli are located, along with parts of the renal tubules. So when we talk about filtration, we are mainly referring to the processes that begin in the cortex. Filtration starts here because this is where blood first enters the glomerular capillaries. So moving inwards, we have the medulla. So these regions appear darker and is organized into renal pyramids. The loops of Henle and the collecting ducts extend down into this region. The medulla plays a key role in concentrating urine because it maintains a gradient of osmolarity that allows water to be reabsorbed when needed. And at the center, we have the renal pelvis. So this is not involved in filtration or reabsorption. Instead, it acts as a collecting chamber. So urine formed in the nephrons drains into minor calyces, then major calyces, and finally into the renal pelvis before flowing into the ureter. So at the gross level, cortex is where filtration begins, the medulla is where concentration is established, and the renal pelvis is where urine is collected before leaving the kidney. Okay, now we zoom into the functional unit of the kidney, which is the nephron. So each kidney contains about 1 to 1.2 million nephron per kidney. But this number is highest in early adulthood and gradually decreases with age, especially after around 40 to 45 years old. So as nephron number declines, the overall functional reserve of the kidney also decreases. So that is one reason kidney function tends to decline gradually with aging, even in otherwise healthy individuals. So each nephron has two main components, the renal corpuscle and the renal tubules. The renal corpuscle is where filtration takes place. So this is the starting point of urine formation. And then the renal tubule modifies the filtrates. It reabsorbs substances that the body needs and secretes others that needs to be removed. So you can think of the nephron as having two main steps. First, filtration at the corpuscle. Second, processing and adjustment along the tubule. Alright, so now let's take a look at each of these structures in more detail. Okay, let us first focus on the renal corpuscle. The renal corpuscle consists of two main parts, the glomerulus and the Bowman's capsules. The glomerulus is a tub of capillaries. What makes it unique is that it is connected to two arterioles. Blood enters the glomerular capillaries through the afferent arterioles and leaves through the efferent arteriole. So this arrangement helps maintain a relatively high pressure within the glomerulus, which is necessary for filtration. And surrounding the glomerulus is Bowman's capsule. Bowman's capsule, it has two layers, an outer parietal layer and an inner visceral layer. The visceral layer is made up of specialized cells called podocytes. So these podocytes form part of the filtration barrier, and it plays a key role in determining what can pass into the filtrate. So this entire structure is the site of filtration. So as blood flows through the glomerular capillaries, part of the plasma is filtered across the filtration membrane, into the space between the two layers of Bowman's capsule. So this space is called Bowman's space. The fluid that collects there is the glomerular filtrate. So from Bowman's space, the filtrate flows into the renal tubule for further processing. The portion of blood that is not filtered continues out of the glomerulus through the efferent arteriole. So in summary, the renal corpuscle is the starting point of urine formation. Blood enters, and then plasma is filtered, filtrate enters the tubule, and the remaining blood exits to continue to the systemic circulation. So after filtration occurs in the renal corpuscle, the filtrate enters the renal tubule. The first segment it enters is the proximal tubule. So the proximal tubule has two parts, a coiled segment called the proximal convoluted tubule, followed by a straight segment known as the proximal straight tubule. So most of this segment lies within the renal cortex. So functionally, the proximal tubule is extremely important. This is where bulk reabsorption takes place. About 2/3 of the filtered water and sodium are reabsorbed here. And almost all filtered glucose and amino acids are reabsorbed here as well under normal conditions. So although filtration happens at the glomerulus, the proximal tubule is where the body starts reclaiming back what it needs. So if this segment does not function properly, we lose essential substances in the urine. And then after leaving the proximal tubule, the filtrate enters the loop of Henle. The loop of Henle is shaped like a U and extends from the cortex down into the medulla and then back up again. It has three functional parts: the thin descending limb, thin ascending limb, and the thick ascending limb. Each part behaves differently. The descending limb is more permeable to water, while the ascending limb is less permeable to water, but actively transport ions. So because of this difference, the loop of Henle plays a key role in establishing the osmotic gradient in the medulla. The medullary gradient is essential for the kidney's ability to concentrate urine. Without it, the kidney would not be able to conserve water effectively during dehydration. So in simple terms, the proximal tubule reclaims most of what was filtered, and then the loop of Henle set up the condition needed for urine concentration. So after the loop of Henle, the filtrate enters the distal tubule. The distal tubule lies mainly within the renal cortex. But compared to the proximal tubule, it handles a smaller volume of fluid, but its role is very important. This is where fine-tuning takes place. In the distal tubule, the kidney adjusts electrolyte balance more precisely. Sodium, potassium, calcium, hydrogen ions can be regulated here depending on the body's needs. And hormones such as aldosterone also influence what happens in this segment. So if you think of the proximal tubule as bulk reabsorption, the distal tubule is more about adjustment and control. And after passing through the distal tubule, the filtrate enters the collecting duct. And several collecting ducts join together and eventually form a larger papillary duct which drains into a minor calyx. And from there, urine flows into the renal pelvis and then into the ureter. The collecting ducts plays a crucial role in determining the final urine concentration. So this is where water reabsorption can be increased or decreased depending on the body's hydration status. And then hormones like antidiuretic hormone or ADH, it acts mainly at this level. So by the time the fluid leaves the collecting ducts, its composition has been carefully modified. What began as a large volume of filtrate has now been adjusted into the final urine. So in summary, the proximal tubule handles the reabsorption. The loop of Henle established the medullary gradient. The distal tubule fine-tunes the electrolytes, and the collecting duct determines the final concentration of urine. Near to the corpuscle, there is this structure called juxtaglomerular apparatus. Juxta in Italian means next to or near. So that is why the name is juxtaglomerular, because it is near to the glomerulus. Its exact location is at the junction of the afferent and efferent arterioles, and also a portion of the distal tubule. So this is the juxtaglomerular apparatus. It consists of macula densa. This is macula densa. It acts as sodium sensors in the tubular fluid. And then there is extraglomerular mesangial cells, which help hold the structure of the apparatus, and also juxtaglomerular cells. This is juxtaglomerular cells, whereby the function is to secrete renin, which is a type of hormone. But although this apparatus is relatively small, but it plays a very important role. It helps regulate the function of each nephron. There are actually two types of nephron, cortical and juxtamedullary. About 85% of all nephrons are cortical nephrons, right? These are located mainly in the outer part of the cortex. Their loop of Henle are relatively short and do not extend very deep into the medulla. So cortical nephrons are primarily involved in routine filtration and reabsorption. They filter plasma and reabsorb essential substances like water, glucose, and electrolytes. The remaining 15% are juxtamedullary nephrons. They are located near the junction between the cortex and the medulla. So what makes them special is their long loop of Henle, which extend deep into the medulla. The structural difference from the cortical nephron is very important. The long loop of Henle allow the juxtamedullary nephron to create and maintain the osmotic gradient in the medulla. The gradient is what enables the kidney to concentrate urine when necessary, especially during dehydration. Alright, so although juxtamedullary nephrons are fewer in number, they play a crucial role in water conservation and urine concentration. So basically, cortical nephron carry out most of the filtration, while juxtamedullary nephrons are essential for concentrating urine and maintaining water balance. Okay, now we move on to the renal blood supply. The kidney receives about 20 to 25% of resting cardiac output. So that is a very large proportion of blood flow for a relatively small organ. So why does the kidney need so much blood? Because its primary role is to filter plasma continuously. So high blood flow ensures that large volumes of plasma are delivered to the glomeruli for filtration. So this allows efficient removal of waste products and also tight regulation of fluid and electrolyte balance. So now let us trace the path of blood through the kidney. So blood leaves the abdominal aorta and enters the kidney through the renal artery. And inside the kidney, the renal artery branches into segmental artery and then into interlobar artery, which runs between the renal pyramids. And then these then branch into arcuate arteries, which arch along the boundary between the cortex and medulla. From the arcuate arteries, smaller cortical arteries extend into the cortex. And from there, blood enters the afferent arteriole of each nephron. The afferent arteriole delivers blood into the glomerular capillaries where filtration occurs. And then after filtration, blood leaves the glomerulus through the efferent arteriole. So this is important. Instead of draining into a vein immediately, it goes into another capillary network. So these second capillaries in cortical nephrons, they are called peritubular capillaries. In juxtamedullary nephrons, they are called vasa recta. So these capillaries are mainly involved in reabsorption. So after passing through these capillary networks, blood drains into cortical radiate vein, then into arcuate veins, interlobar vein, and finally into the renal vein. And then the renal vein then returns the blood to the inferior vena cava and back to the heart. So the kidney has a unique arrangement. Blood passes through two capillary beds in series, first for filtration and then for reabsorption before returning to the systemic circulation. In most parts of the body, blood flows from an arteriole into a capillary bed and then directly into a venule. That is the usual pattern. But in the kidney, it is different. In the kidney, blood flows from the afferent arteriole into the glomerular capillaries, then into another arteriole, the efferent arteriole, and then before entering a second capillary network. So the first capillary bed, the glomerulus, operates at relatively high pressure. So this high pressure is essential because it pushes fluid out of the capillaries and into Bowman's space. In other words, it promotes filtration. And after blood leaves the glomerulus, it passes through the efferent arteriole before entering the second capillary bed. This efferent arteriole is very important because its diameter helps determine how much pressure is maintained inside the glomerulus. If the efferent arteriole constricts, blood has more difficulty leaving the glomerulus, so pressure inside the glomerular capillaries increase. So filtration increase. If the efferent arteriole dilate, blood leaves more easily. So the pressure inside the glomerular reduce, filtration also reduce. So the efferent arteriole act as a pressure regulator between the two capillary beds. And then once blood enters the peritubular capillaries, the pressure is much lower. So this low pressure promotes reabsorption. So fluids and solutes that were filtered into the tubules can now move back into the bloodstream. So the arrangement is not accidental. It is designed so that the first capillary bed promotes filtration and the second capillary bed promotes reabsorption. And the efferent arteriole helps control the balance between the two. So this double capillary system is unique to the kidney. Okay, now that we understand the structure and blood supply, let us focus on filtration itself. So glomerular filtration is the very first step in urine formation. It is the point where blood and urine pathways begin to separate. So as blood flows through the glomerular capillaries, part of the plasma is pushed across the filtration membrane and into Bowman's space. What is being filtered is not whole blood. Red blood cell, white blood cells, and platelets remain inside the capillaries. And large plasma proteins are also mostly retained in the blood. What crosses Bowman's space is mainly water and small dissolved substances. So the fluid that collects there is called the glomerular filtrate. At this stage, the filtrate is quite similar to plasma in terms of small solutes. It contains water, sodium, potassium, chloride, glucose, amino acids, urea, and many other small solutes. The key difference is that it contains almost no proteins and no blood cells. So you can think of glomerular filtration as producing a protein-free plasma. So now the question is, what determines how much gets filtered? Filtration depends on two major factors. First, the properties of the filtration membrane, its structure, permeability, as well as selectivity. Second, the pressure difference across the glomerular capillary wall, what we call as Starling forces. So filtration is controlled both by the physical characteristic of the barrier and also by the force pushing fluid across it. Alright, so let's look at closely at these two factors. Now let's take a look at the filtration membrane. The filtration membrane has three distinct layers, and each layer plays a role in determining what can pass from the blood into Bowman's space. The first layer is the fenestrated capillary endothelium. So these endothelial cells have small pores or fenestrations. So these openings allow water and small solids to pass through easily, but they are small enough to prevent blood cells from crossing. So under normal condition, red blood cell and other form elements remain inside the capillaries. The second layer is the glomerular basement membrane. This is a thick, specialized layer of extracellular matrix located between the endothelium and the podocytes. It acts as a major barrier to large molecules, especially plasma proteins. The third layer is the podocyte slit diaphragm. Podocytes are specialized epithelial cells that wrap around the glomerular capillaries. Their food processes interlock, so that there is only narrow gaps between them. So these gaps are covered by a thin diaphragm that acts as the final filtration barrier. So when plasma is filtered, it must pass through all these three layers. First the endothelium, then the basement membrane, and finally the slit diaphragm. So together, these layers ensure that water and small solids are filtered, white cells and proteins are retained in the bloodstream. If any part of this barrier is damaged, its selectivity is lost. For example, if the basement membrane or podocytes are disrupted, proteins can leak into the urine. So this condition is known as proteinuria and is often a sign of glomerular disease. So the filtration membrane is not just a simple filter. It is specially designed to let small substances pass while keeping important proteins in the blood. Now, the ability of a substance to cross the filtration membrane depends mainly on two factors, molecular size and ionic charge. Size is the primary determinant. In general, the smaller the molecule, the more easily it can pass through the filtration barrier. Water, glucose, sodium, potassium chloride, and other small solutes are freely filtered. So these molecules are small enough to pass through all the three layers of the filtration membrane without significant restriction. Small proteins like myoglobins may also be filtered to some extent, but they are relatively small in size. But larger proteins like albumin are retained in the plasma because they are too large to pass through the filtration barrier. And charge also plays an important role. The filtration membrane, especially the basement membrane and podocyte layer, carries a negative charge. Many plasma proteins, including albumin, are also negatively charged. So because they are both negative and negative, so they repel each other. This ionic charge and so they are less likely to cross the membrane. On the other hand, positively charged molecules or cationic are filtered more easily because they are not repelled by the negatively charged membrane. So when we think about filtration, we need to remember two things. First, size determines whether a molecule can pass at all. And secondly, charge influences how easily it passes. So both factors work together to ensure that useful small solids are filtered while large and important proteins are retained in the bloodstream. Aside from the filtration membrane, glomerular filtration also depend on pressure differences. So these pressures are known as Stirling forces. Filtration only happens because there is a driving force pushes fluid out of the glomerular capillaries into Bowman space. But at the same time, there are forces that resist this movement. So what we are looking for at is a balance of pressures. So there are three main pressures. Glomerular hydrostatic pressure or PGC. So this is the pressure exerted by the blood inside the glomerular capillaries. Because the glomerulus is supplied directly by an arteriole and drains into another arteriole, so the pressure here is quite high compared to most other capillary beds. So the high pressure pushes fluid out of the capillaries and into Bowman space. So this is the main force that favors filtration. The second pressure is plasma oncotic pressure, or written as PIGC. So this is generated by plasma protein, mainly albumin, within the glomerular capillaries. Proteins cannot cross the filtration membrane easily, so they remain in the blood, right? So this protein attract water. So at the same time, it pulls fluid back into the capillary. So plasma protein or plasma oncotic pressure oppose filtration. The third pressure is Bowman's capsule hydrostatic pressure or BBC. So this is the pressure exerted by the fluid that has already entered the Bowman space. So as fluid accumulates here, it creates a backward pressure that resists further movement of fluid into the capsule. So this pressure also opposes filtration. So if you simplify it, only one force push fluid out and two force push the fluid back. So the overall effect of these three pressures give us what we call the net filtration pressure or NFP. So what is the net filtration pressure? The net filtration pressure is the final driving force that determines whether fluid will move out of the glomerular capillaries into Bowman space. So mathematically, it is calculated as glomerular hydrostatic pressure minus the sum of plasma oncotic pressure and Bowman's capsule pressure. So glomerular hydrostatic pressure is approximately 60 millimeter mercury. Plasma oncotic pressure is about 32 millimeter mercury, and Bowman's capsule pressure is about 18 millimeter mercury. So using this equation, the net filtration pressure is roughly about 10 millimeter mercury. So even though 10 millimeter mercury sounds small, but remember that the glomerular capillaries have a large surface area, so that is why a small pressure can still produce a high filtration rate. Now let us talk about glomerular filtration rate or GFR. So GFR refers to the total volume of filtrate formed by all the glomeruli in both kidneys per minute. In simple terms, it tells us how much plasma is being filtered by the kidneys every minute. In a healthy adult, GFR is about 125 milliliter per minute. And if you calculate that over 24 hours, that comes up to roughly about 180 liter per day. Now, think about that number for a moment. We filter about 180 liters of fluid every day, but we only produce about 1 to 2 liters of urine. So what happens to the rest? More than 99% of the filtered fluid is reabsorbed along the renal tubules. So this tells us how important tubular reabsorption is. Filtration is just the first step. Most of what is filtered is actually taken back into the body as needed. GFR is slightly higher in males compared to females, and it is influenced by body size. A larger individual with a larger body surface area may have a slightly higher GFR because there is more tissue to supply and more metabolic waste to handle. Now, why is it important to maintain a relatively stable GFR? If GFR increases too much, fluid moves too quickly across the nephron. So the tubule may not have enough time to reabsorb water and essential solids properly. So this can lead to excessive loss of substances that the body actually needs. And on the other hand, if GFR decreases too much, filtration becomes insufficient. Waste product like urea and creatinine will begin to accumulate in the blood. So that is when we start seeing signs of renal impairment. So maintaining a stable GFR is crucial for maintaining fluid balance, electrolyte balance, and also removal of metabolic waste. So in other words, GFR is one of the key indicators of overall kidney function. And clinically, when we assess kidney function, what we are really estimating is GFR. A reduced GFR often signs early kidney disease even before symptoms appear. So GFR is not just a number, it is a reflection of how effectively the kidneys are filtering plasma and maintaining the internal balance. Okay, now that we understand what GFR is, the next question is what determines it? GFR depends on two main factors, the net filtration pressure and the filtration coefficient. We have already discussed the net filtration pressure. It is determined by the three Starling forces: glomerular hydrostatic pressure, plasma oncotic pressure, and Bowman's capsule pressure. So that tells us how strong the driving force for filtration is. The second factor is the filtration coefficient, or Kf. So what exactly is Kf? It reflects the physical properties of the filtration barrier, which is the three layers of the filtration membrane. It depends on two things: the hydraulic conductivity of the membrane, which means how easily water can pass through it, and the surface area available for filtration. In simple terms, Kf tells us how permeable the membrane is and how large the filtering surface is. The normal value of Kf is about 12.5 milliliter per minute per millimeter mercury. If Kf increases, that means more fluid can pass through the membrane. So GFR increase. And vice versa, if Kf decrease, filtration becomes less efficient, so GFR decrease. So if we put it together, GFR depends on two things. First, how strong the driving pressure is, that is the net filtration pressure, and secondly, how permeable and how large the filter is, so that is a filtration coefficient. Anything that changes the pressure inside the glomerulus or alters the structure of the filtration membrane will ultimately change GFR value. For example, changes in blood flow can alter glomerular hydrostatic pressure, or damage to the filtration membrane, such as in glomerular disease, can reduce surface area or alter the permeability of the membrane. So both of these situations can affect GFR. So when we talk about changes in GFR, we are really talking about changes in pressure, structure, or both. So, okay, that brings us to the end of this lesson. We started with the structure of the nephron and how it supports its function. And then we saw how the kidney's unique blood supply allow high pressure filtration followed by reabsorption. And then we also discussed how plasma is filtered across a specialized membrane, and also how Starling forces determine the net filtration pressure. And then finally, we define GFR and its use in estimating the effectiveness of kidney function. So with that, thank you.